Hi everyone, my name is Yue. I'm a current PhD student from the University of Texas at Austin. So in my today's talk, I'm gonna show you my current work, which is coordinate translational efficiency is a functional organizing principle of mammalian transcripts across cell types. So first of all, I want to refresh you about concept that gene with similar mRNA expression pattern, they also share same biological function. So as you can see with this cartoon that gene A and gene B, they have similar mRNA expression across different conditions. So the conditions can be different treatments, drug treatments, or knockdown, knockout. And as you can see, since they have similar mRNA expression pattern, normally people find they also have shared biological function. And people called these two genes a co-expressed genes. However, when we have another gene, it has different mRNA expression pattern and is not co-expressed with gene A and B. And people find normally this gene also has some other biological function that not related to gene A and B. So this has been widely used for people to identify novel gene functions, mRNA regulation. But we were wondering, what about genes that shared similar translational efficiency pattern? And this, thanks to our surprise, has never been explored before. So one of the key reasons may be because that we lack of enough data source for us to really capture the expression pattern in this translational efficiency level. And do genes that with this similar translational efficiency pattern also have the same biological function? And do genes with similar translational efficiency that they are conserved between different species? These are the questions that I like to answer with my project. So in order to answer all this information, what we need to know first is the technology that can capture translation efficiency. And this can actually be measured with ribosome profiling. And I'm not sure how many people are familiar with this technology, but it's quite similar to RNA-seq, which is the next generation sequencing technology that capture the genome-wide mRNA level that are under translation. So as you can see in this technology, we measure which mRNA are protected by the ribosome. And we treat with nuclease and really get the ribosome protected footprints and do the site selection, and then we do the sequencing. This is the method that how we get the ribosome bonded footprints. And we further normalize this information with the actually mRNA expression level, then we get the translation efficiency level, aka we call it TE. And this is the canonical method for us to actually understand the speed, the efficiency of translation. And it also gives us a chance to really measure that which gene has a higher TE and which gene has a lower TE. And if you still remember, the goal of my project is really to understand the TE similarity pattern across different conditions for all the genes. Then what we need to have is a robust database for us to capture the ribosome profiling data source. And luckily, we have the data from GLBIO, but there's still some disadvantages from this data source. So the first thing is the meta information is really a mess. So like there's a lot of missing information, at least for ribosome profiling data, such as the cell line we found is not matched with the literature. And there are some things that are not really filled in the database. And you need to do the literature check. And the most important thing that I want to highlight here is there is no matching information between ribosome profiling and RNA-seq in GEO. If you still remember the formula that how we calculate TE, you need to have both of these two information for a specific sample, and then you can get the TE score. So if you don't have this information from the GEO, then you will need to double check from the literature and then get this information to calculate the TE, and then to really capture the TE similarity pattern across genes. So here is the evaluation results from our ribosome database. So actually, we did a manual curation matching between ribosome profiling data and RNA-seq data. So as you can see in this box plot, the y-axis is the R square, and the yellow one means that these are the matching information that you can get from our Rebel base. And we also did random matching between ribosome profiling data versus RNA-seq data from the same study from GEO. And you can see that clearly that we have a higher R square than the random matching from the same study. And here the gray boxes indicate that we did random matching between the ribosome profiling with RNA-seq from other studies. So in this way, you can see that the matching score is quite low. And besides that, in order to uniformly compare all the data, we reprocess all the sequencing data from GO for ribosome profiling and have this uniform analysis results. And here I also want to highlight something that we're missing from the currently available ribosome profiling data source. One thing is they have the TE data, and another thing is what they analyze the ribosome profiling data. Is actually related to ribosome protected footprint. So normally people just use the hard cutoff, use a specific range to extract the ribosome protected footprint from the sequencing data. As you can see from this heat map, this is all the human ribosome profiling data from GEO. If you still remember the workflow from ribosome profiling, that the footprint length may vary based on the after you treat with the enzyme. So most of the sample treat with RNAs. So if you actually treat with RNAs, the length distribution should be really narrow, around 28 to 32. But there's still some protocols that are using the mRNAs as the treatment. So with that kind of data source, if you really dig into the real length distribution from GEO, you will see that they have a wider read counts distribution. So if we just use a really hard cutoff for all the samples, then you will miss a lot of useful information. So after we did all this data processing, now we have thousands of human and mouse data available for us to really understand TE similarity pattern across species and genes. And there's another, I think, very interesting thing that I want to highlight is actually we also did a secondary analysis for the published data source, and we found that only 80% of them are have a really high quality. So for one thing is about the CDI's percentage. So as you can see that since the ribosome profiling is trying to measure which mRNA is under translation, so we think that most of the reads should come from CDS region if they are under translation. But as you can see, there's still a lot of samples, they have really low CDS coverage, meaning that they have really low quality when they're doing the sequencing. And another thing is we found a lot of samples, as you can see, they don't have really good coverage, indicating if you want to really understand the TE similarity pattern as many genes as possible, then if the genes, if the sample have really low coverage, meaning that you will not get enough gene coverage for the total analysis. So as you can see, though there are the existing published data, there's still a lot of information are not really suitable for large-scale data analysis. So we think when we do the metadata analysis, large-scale data analysis from existing data, it should be really careful about data source that we were using. And after that is the step to really calculate the TE. Instead of using the canonical way, we also use another method, which is a linear regression-based model to actually use the residual as the TE. So in this method, instead of using the ribosome profiling directly divided by RNA-seq, we try to avoid the unwanted spurious correlation between TE and RNA-seq signal. And then we want to see how informative of our TE score with the actually biological information. Then we currently TE with the proteomics data for 7 cell lines, compare this with the canonical way to calculate TE. So as you can see, with our method, we have higher Spearman correlation with this method versus the proteomics data, indicating this method preserves more biological information. And then we try to see the correlation between the human TE versus mouse TE. And to our surprise, actually in this TE level, there's like really high conservation between human and TE, human and mouse homologous gene. But we still find some outlier genes, they have higher TE in human, in either human and mouse. We try to do an enrichment analysis to see what kind of biological functions these genes are enriched in. So as you can see, for genes that have higher TE score in human, and they are related to RNA binding function, and for the genes that have high TE scores in mouse, they are actually related to the extracellular matrix function. And the reason we think is that for the ribosome profiling data from the current data source, most of the mouse data, they are researching on development. And the extracellular matrix is quite essential for the earlier mouse development. And then it's a time to really calculate the TE similarity pattern among genes. So here I want to show you an example from the current data source. And there's 4 genes. So these 3 RPS genes, as you can see, they have a quite similar TE pattern, as you can see, based on the color, of course, these human cell lines and tissues. And then we also introduced another gene as an example, HK2. It has different TE pattern across these conditions. And we calculate the similarity pattern with a proportionality score. It's quite similar to Spearman and Pearson correlation, but only measure the data in proportional level. And then we can see that these 3 RPS genes, they have a relative high row score, indicating that they have a high TE similarity pattern. On the other hand, HK2, it shows different pattern and also have quite low row score with the 3 RPS genes. And for the detailed biological function, the 3 RPS genes, they are actually small ribosome subunit proteins, and HK2, on the other hand, is a glycolytic related genes. And we also try to see the TE similarity score pattern among homologous gene pairs, how are they conserved between human and mouse. So we try to plot the row score for each gene pairs with their mouse level and human level. And in this plot, we aggregate the risk count, the similarity score count for within certain range, and do a log transform, and then color them with the different color and size. And then, as you can see that in this level, the homologous genes pairs is TE similarity pattern are also conserved between human and mouse. And then the last question we want to answer is how genes associate with same biological function. Do they have similar translational efficiency pattern? We evaluate this with our neighbor working strategy and evaluate with AURC score. So in this strategy, that if genes fall within specific biological function, if they have high similarity pattern with each other, then the AURC score is much higher than 0.5. If they do not show a strong similarity TE pattern, then the AURC score is around 0.5 or even lower. Then we test thousands of human and mouse data, and the median AURC score is much higher than 0.5, indicating that genes associate with same biological functions, that they have higher TE similarity pattern. And one interesting thing that we found genes with specific biological function, such as this MAP kinase activity, the gene within them, they only show strong TE similarity pattern, but they are not really co-expressed. And this is not the only case that we find a lot of biological function, they show strong regulation in TE level instead of mRNA regulation level. So in summary, in today's talk, first of all, I show you that how we calculate translational efficiency across different conditions with the current data source, and the TE is conserved between human and mouse. And the next thing is gene with similarity translational efficiency pattern also share same biological function. and some of them may not be co-expressed in mRNA level. And at last, I want to thank all the people in my lab, especially my PI and our collaborator from Sanofi. They're using our score to do a machine learning, deep learning prediction to optimize the mRNA vaccine development, and also the funding source and also the support from the travel fellowship from the conference. So that's all. Thank you.
So you use Go terms as your definition of protein function.
Mm-hmm.
I would have guessed that in the past, RNA-seq or RNA expression patterns were used to predict or annotate protein functions.
You're asking how about like in co-expression?
I would guess that yes, that
they're
not for
like
translational efficiency, but that the reason they agree for expression is probably part of the annotation process. I'm guessing those aren't independent.
Yes, I think there's some, like based on the evaluation for co-expression, like a lot of them, like they co-express together, they from the same Go term. And
but no, what I want to know is, did people assign the Go term if they were co-expressed?
Most of them, most of them are yes. So there's another part I didn't show with we also did co-expression with the data source we have. Like the co-expression score is a little bit higher than TE, like the enrichment. So yeah.
Yeah, no, what I'm trying to suggest is that it's not really an interesting result with the RNA-seq if they use that to get the functions in the first place.
Yes.
So in some sense, it seems like your most important result is where TE does something different, because that's something that wasn't used to get the annotations in the first place.
Yes, yes. We just want to see that there's still some part they're not got directly from co-expression, I mean the Go term. And we found that there's a lot of regulation. Some of the regulation, they're more enriched in TE. And what we want to do is trying to predict the novel gene function with this kind of information. Are there any regulation just occur in the TE level?
It's a nice piece of work.
Thank you. Thank you.